Assalamualaikum everyone, my name is Nurul Imandayana binti Adnan, and today I'll be presenting my research on the analysis of antimicrobial resistant gene trends of Pseudomonas aeruginosa whole genome sequences in Malaysia, which are guided by Dr. Muhammad Izwan bin Ismail and Dr. Muhammad Naziz bin Saad. Pseudomonas aeruginosa is a gram-negative aerobic non-spore-forming rod-shaped bacterium which has a complex cell wall, making it difficult for antibiotics to penetrate. P. aeruginosa is one of the important escape pathogens that is known for causing hospital-associated infections and its biofilm formation through quorum sensing, which has high adaptability towards its surroundings. P. aeruginosa can be treated using the 6 classes of antibiotics, but it has slowly started to build resistance against them. And this is called the antimicrobial resistance of P. aeruginosa, where P. aeruginosa no longer respond effectively to antimicrobial medicines. And in this study, whole genome sequencing (WGS) is used to detect the resistant gene in P. aeruginosa. According to the NSAR 2024 report in Malaysia, the resistant rates of P. aeruginosa towards key antibiotics like imipenem and meropenem have shown an increase from 2023 to 2024, which brings me to the research question, which is what are the trends and patterns of antimicrobial resistance in P. aeruginosa? Are there diverse genomic characteristics of P. aeruginosa strains? And which antimicrobial resistant genes are present in P. aeruginosa strains isolated in Malaysia between 2010 to 2024? And in order to answer all these questions, I have come up with these 3 objectives, which is to compile and curate the antimicrobial resistance data of P. aeruginosa between 2010 to 2024 from the National Surveillance Antimicrobial Report, NSAR, to perform bioinformatics analysis on the genome sequences of P. aeruginosa Malaysian strains using NCBI, and to analyze the antimicrobial resistant genes of P. aeruginosa using CARD and ResFinder. This study is significant as the AMR of P. aeruginosa are inconsistently increasing and decreasing over the years. It may cause treatments to not be as effective as before. Hence, by tracking the new clones and new emerging resistance strains, this study can provide insights for healthcare professionals to develop more effective infection control strategies. The first theory that is going to be applied in this thesis is antimicrobial resistance, AMR, that occurs when microorganisms build resistance against antibiotics because of genetic changes that occur when microorganisms are exposed to stress. Resistance can develop through several mechanisms, including enzyme modification, alteration of antibiotic target sites, efflux pumps, and horizontal gene transfer, where resistant genes are shared between bacteria. The second theory is Pseudomonas aeruginosa, where Pseudomonas aeruginosa is commonly found in soil, plant roots, and aquatic environments. It can infect both healthy individuals and those with weakened immune systems. The bacterium is highly virulent due to toxin secretion, virulence regulation strategies, and strong biofilm forming ability. It can cause infections such as pneumonia, bloodstream infections, surgical site infections, and many more. Next is whole genome sequencing, WGS. WGS is a type of next generation sequencing, NGS, that allows sequencing of an organism's entire genome. Examples of other sequencing technologies are first generation sequencing, long read sequencing, and shotgun sequencing. Through WGS, the antimicrobial resistant genes can be detected as it can differentiate between active and passive genes in the genome. Previous studies have the same view on the topic, which is first, P. aeruginosa is a multi-drug resistant pathogen. Second, they highlighted AMR as a critical health threat or concern to society. Third, resistant rates and patterns of AMR vary depending on region or other contexts. Based on the graph from the NSAR report 2024, it can be seen that P. aeruginosa AMR has an inconsistency throughout the years, but there is still a knowledge gap on the formation of resistant genes in P. aeruginosa and the emergence of antimicrobial resistance of P. aeruginosa. The study design of this thesis is the database analysis of antimicrobial resistant gene trends of Pseudomonas aeruginosa in Malaysia, and the data are gathered from the National Surveillance Antimicrobial Report, NSAR, and the NCBI database. Bioinformatics tools used in this study is CARD and ResFinder. Both of these is used to list the resistant genes present in the genome sequences. SecHub is used to annotate the AMR gene sequences. Eugene will be used to clean and prepare the sequences for further analysis. And Microsoft Excel is used to curate and analyze trends and timelines. The methodology of this study starts with compiling and curating the data from NSAR and observe the trends and timelines of the AMR of P. aeruginosa. Then, data of P. aeruginosa gene sequences will be collected from NCBI. Next, all the gene sequences will be screened for AMR genes in CARD and ResFinder. Furthermore, the retrieval of AMR genes starts, and in this step, SecHub and UGene will be used. Moreover, the AMR genes will be sorted after in Microsoft Excel for further analysis. The last step is the analysis of trends and timelines of the resistance genes of P. aeruginosa. The expected outcomes of this research are as follows. First, the overall resistance rates of P. aeruginosa in Malaysia are determined. Second, this study can identify variable resistant trends of P. aeruginosa across different states and years. Lastly, the different resistant genes present in P. aeruginosa strains over time are able to be predicted. And these are the graphs that are similar to what is going to be in my results. However, there are some limitations in this study, such as an inconsistent data. The data collected for a year may not be the same amount as the previous year. Next, the inaccurate sequence screening. There may be errors in the screening of resistant genes from the sequences. Moreover, the risk that may be faced in the future is human errors, such as errors in computing results and computational limitations, such as computational resources that may be limited. To address these challenges, I will implement some mitigation strategies. First, weekly meetings with my supervisor will be held to verify data and ensure the analysis is on track. Second, reliable online tools will be used to cross-check and validate the data. This is my Gantt chart for next semester.